I think it's important that you understand, in particular, because when people think about the extracellular matrix, and oftentimes when people use the ECM or study the ECM in their own research, there are a lot of there are some key points I think that are overlooked. So I want to emphasize those today. So the first thing is, what is the ECM? And it's really the non-cellular component of animal tissues and organs. You're going to have a lecture tomorrow from Charlie Anderson at Penn State, who's going to tell you about the plant cell wall. And I think it would be very, very helpful if you tried to, think in your mind about a comparison between the two. CEMB, a bunch of us are including faculty and trainees, are in the process of writing a review comparing the plant cell wall and the animal ECM. And it's very interesting what's similar and what's different. So I think it would be helpful for you if you thought about that tomorrow. But it is the non-cellular component of animal tissues and organs, and it's a hydrated polymer network. Some of you have a polymer lab today, and it would be worth thinking about the ECM in that context. So it contains fibrous proteins that are there for structure and polysaccharides that are space filling, and of course, a ton of water. And the other key thing to keep in mind is that it's really crowded. Like the inside of the cell that is incredibly crowded, the ECM is also very crowded. And I just included you a couple of actual images or drawings of the ECM, but this has major import when you think about cells moving through the ECM, solutes moving through the ECM, things getting liquids moving through the ECM, and organization of the ECM, the fact that it is just so crowded. I think oftentimes in pictures, people draw a cell and they have a couple little wisps of collagen around it. Totally not realistic. It's a packed space, both inside and outside of the cell. So the other thing I want, and I just sort of show this as an illustrative picture, I want you to appreciate just how complex the components of the ECM are. So this is from one of the first papers from something called the Matrosome Project that was led by Alexandra Naba, who was a postdoc at the time in Richard Heinz's lab at MIT. And what she developed was a combined proteomics and bioinformatics approach to identify every proteinaceous component in the ECM. So this is only protein. It didn't include proteoglycan or glycosaminoglycans. And the first thing I want you to note is that there's a ton of stuff. We think about the ECM as, collagen and a couple other fluffy things. But in fact, if you look at the numbers of proteins, secreted factors, regulators in either the human Homo sapiens or mouse muscularis, you can see that there are hundreds of different kinds of components of the ECM. And they can be roughly divided into a couple different ways. The first is there are the core matrosome components, and these are the proteins and that we're going to talk about today. Things like the big glycoproteins, the structural glycoproteins like collagen and elastin, and the proteoglycans that are the space fillers. But there's also a second category of these matrosome associated proteins. They may include soluble factors that are binding to the ECM. They may be cross-linkers or other kinds of regulators or proteases that determine the function or the structure of the ECM. So that's so there's a ton of different components of the ECM. There are these core components and then these associated components. You can also divide it in another way into thinking about two categories of the ECM. There is the basement membrane and then the interstitial matrix. So the basement membrane is a sheet-like component of the ECM, and it holds epithelial cells. So epithelial cells, just to remind you, are the lining or coating cells of the body. So, inside your mouth, on your skin, etc. And they function in sheets, and they have to adhere to something. They're polarized, they have a top and a bottom, and so they adhere to the basement membrane. So that is one category of matrosome components. The other category is interstitial, and this is the part of the ECM that is between cells and tissues. It has the gel-like polysaccharides and the fibrous proteins, and it supports fluid flow. I have also included in this category the glycocalyx. I'm not sure everyone would agree with that, but no one ever talks about the glycocalyx in the context of the ECM, and I think it's important. So the glycocalyx is this carbohydrate and matrix protein-rich layer on the outside of cells. So we all think about, a little growth factor zipping in, bang, 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 sees a cell receptor on the outside, binds to it, and there's signaling. But in reality, your little growth factor is zipping along and encounters this incredibly charged and dense layer of matrix proteins and carbohydrates on the outside of a cell. It can be, the diameter of the glycocalyx can be as wide as the cell. It can be the same as the cell itself. So I'm going to show you some pictures at the end of the lecture. But so the point is, it's a complex, the matrix is complex. There are core components and associated components, and it can be roughly divided into the basement membrane matrix and the interstitial matrix. So there are four key things that I want you to know about the ECM and to think about it as I go through the rest of the lecture. The first is that it's very heterogeneous. So if you look at composition of different tissues as far as the ECM, you can see 2 or more orders of magnitude between tissues. As an example, your brain has little to no collagen, but your tendons are almost all collagen. And if you look at other matrix components, it's just amazing how different tissues are in as far as how much and what ECM they have. It's also incredibly dynamic. So not only is it different in different tissues, but the ECM changes at different stages of development. It changes in diseases. And these differences include things like post-translational modifications. So how the ECM is getting connected to itself and to other components of the ECM, how it's organized and what it's composed of. So, we all for experiments, we often take some collagen that we purchase from somewhere that we don't necessarily know the structure of very well, and consider that to represent the ECM. And that's a really, really reductionist view because there's a lot of variability and dynamism in the ECM. The next thing is that it determines physical and biochemical properties of tissues. So the ECM has both tensile and compressive strength, and it's elastic. It has the capability to retain water and to buffer. It's sort of the body buffer. But it also has a significant role in signaling. So the ECM used to be considered sort of structural and inert. And now we know that it plays, the ECM is not only structural and it holds on to growth factors, so it sequesters growth factors, but ECM molecules themselves interact with receptors. You heard a little bit of that from the first talk today, and can serve as signaling molecules. So these are really key things to think about the ECM. I also just want to make this one point that the ECM is being remodeled all the time. So the stromal cells, which are mostly the cells that are making the ECM, is constantly remodeling the ECM just to keep it in equilibrium. So it's sort of, the concept is dynamic equilibrium, because the ECM is constantly subject to stress. And so these stromal cells are constantly remodeling to bring it to a point of equilibrium and homeostasis. If there's a disruption, fibroblasts are going to reorganize their cytoskeleton to return to the baseline. So this is another area where the ECM is really quite dynamic. And this is sort of at a small level happening all the time. So of the ECM molecules, I think I sort of hinted at this before. There are 2 groups. The first is the fibrous proteins, and that includes the collagens, linker proteins like fibronectin and laminin. Laminin is a linker protein for the basement membrane, and elastin. And then the second category are proteoglycans and glycosaminoglycans. And that can be roughly divided into hyaluronic acid, large proteoglycans, and small proteoglycans. And we'll go into a little bit of detail on all of those. So this is I hate this slide, but every time I have to give a lecture on the ECM, I try to find an alternative and have thus far failed, including yesterday. So I just want to give you some sense of the scale of matrix components. And what I want you to take away is that they're really big. So if you look at this middle panel, this is just a graphical representation of a globular protein at the top. And then you can see collagen here in the middle, and you can see it's a lot bigger than a globular protein. But then you look at high hyaluronic acid, and we'll talk about HA a little bit later, but HA swells, it holds water, it's massive. And it can sort of fill a cube with a 300 nanometers on a side, a molecule of hyaluronic acid. Now, if you look on the right, this gives the size of various cells, but it's variable, but generally a cell is about 10 microns on a side. So clearly matrix components are much smaller than cells, but it's a couple orders of magnitude. It's not as large as you might have guessed to start. So one of the things about CEMB is we try to think about time and length scales. And so I want to emphasize for you that you should be thinking about the length scales of a cell, of different matrix components of proteins, how they all fit together. And so just to emphasize that the matrix molecules are bigger than you think, particularly the proteoglycans and glycosaminoglycans. So let's start talking about the collagens. So there are 28 different kinds of collagen. I checked today and it's still 28. And they make up 25 to 30% of the protein mass in your body. It's the most abundant fibrous protein in your body. About 90% of that is type 1 collagen. And that's mostly what we're going to talk about today. And that's mostly what people who study collagen are studying, or people who use collagen are using. So this is, I just put up this figure. So these are just different ways of thinking about the structure of collagen. And one might be more or less helpful to you, depending on how you like to look at, how you like to think about chemical structure. But the major collagens, all of them, all 28, contain what are called the collagen domains. Type 1 collagen is almost all these collagen domains. Some collagens have, much smaller collagen domains. But these domains are made up of gly-XY repeats, where the X is a proline, and the Y is typically a hydroxyproline. And the N of these repeats is in the hundreds to thousands. And so 3 chains of these gly-XY repeats will form a rigid triple helix. And you can see in D down there how what the interactions are between these the three chains in order to form this helix. But they're really tightly packed due to this glycine, which, as you know, is a small amino acid. And they're stabilized by the hydroxyproline and also hydroxylysine residues. So you end up with these really tightly packed and rigid triple helices, just on the basis of the chemistry of these gly-XY repeats. There are multiple families, and I'll show you that in a second. And it depends, the different families of proteins, collagens, and the different collagens have different kinds of collagenous versus non-collagenous domain organizations. And there's also a ton of post-translational modification that is very important for the higher order organization of collagens. So this is just cartoons showing you some of the different families. I think we probably all know about the major fibrillar collagens, type 1, type 2, type 3, and type 5, that form these rigid rods. These are all, triple helices that come together. But there are all these other collagens, and you can see in blue the non-collagenous domains. And in some of these collagens, they're really quite significant, and they determine the formation of all these elaborate structures. So for example, the network forming collagens like collagen 4 and collagen 6 over here they're very important in making the basement membrane, just because they're able to form these networks. The facets bind to the fibrillar collagens and so on. So what I want you to take away from this is that the mechanics of different collagens are going to be different. And they're going to have very different capabilities of binding cells, of binding other matrix components, et cetera, just because the structures are so different. Yeah, go ahead.
Maybe out of like the scope of the presentation, but do you know of any common mutations for collagen that can occur?
So there are a number of different diseases that have mutations in collagen. The one that in different collagens, the one that comes to mind for me, although it's not what I studied, but there's one variant of Ehlers-Danlos syndrome. So Ehlers-Danlos syndrome is a connective tissue disorder where there are a variety of different kinds of mutations, but one group of patients with Ehlers-Danlos has mutations in collagen type 3, and they have various weaknesses in different connective in their connective tissues. In the Ehlers-Danlos, where there's a type 3 collagen mutation, their intestinal wall can be very weak. So, they may be prone to perforation. They may have vascular weakness, and so prone to aneurysms and so on. Other kinds of Ehlers-Danlos are not quite as serious, but people will have very lax ligaments. I mean, they say, I do not have Ehlers-Danlos. I'm like the anti-Ehlers-Danlos, but, if I put my hand like that,
That is a personal question. Like I do have Ehlers-Danlos.
yeah, so you may, if you did this, yeah, it's, yeah, I mean, people can do amazing things, they go like this and their whole hand is divided in half. Yeah, so it's, but there are different kinds. But yeah, they're in different matrix components, absolutely.
Thank you.
Okay. So I just wanted to, I just like this slide because it gives you cute little diagrams, but what the key things are in purple. And this just tells you about collagen biosynthesis. And a couple important things to remember is that biosynthesis occurs both intra and extracellularly. So it starts out with synthesis of the single chains. Collagen 1 is a heterotrimer, so there are two what are called alpha chains, alpha 1 chains and one alpha 2 chain. That isn't so relevant for this talk. And then inside the cell, you have lysine and proline hydroxylation. There may be some O-link glycosylation, and we'll talk about the importance of that in a minute. But you get the formation of these very rigid trimers. And then as I said, the chains form the triple helical procollagens, and they're secreted into the extracellular space. So this is what happens inside the cell. You get the triple helix formed. Outside of the cell, you get, the first thing is that the N and the C propeptides on the collagen chain are cleaved. You can see that here. But one thing to keep in mind is that there are telopeptides on either end of a collagen, and those are retained. So these sort of the collagen chains without their propeptides then self-assemble into fibrils and undergo inter and intramolecular covalent cross-linking. And I'm going to show you another picture of that in a minute. But one thing I want to emphasize is that this O-link glycosylation is really important. And you have these cross-links forming between one of the telopeptide domains and then the interior of the next fibril. Okay, so does that make sense? You can see all of these. There's cross-linking that's related to these telopeptides. One of the reasons I want to bring that up is that you can buy atelocollagen, and you'd sort of want to know if for your experiments, if that's what you have, if you have telocollagen or atelocollagen, because it's going to determine the structure of your collagen. Without the telopeptides, you're not going to have such a complex structure. Okay, but so the point is, inside, you get the formation of the triple helix. Outside, the propeptide domains are cut off. You start to have assembly into fibrils with cross-linking. And then these fibrils with further cross-linking, will form fibers, and these fibers can really be quite large. So this is, when you think of collagen, this is typically the structure that you see, and they form in a staggered way.
A question?
Yes.
Tilo or what? Can you?
Yeah, so let me say,
Can you
so
spell it?
telopeptides, they're just these little domains at the end. And that's where a lot of this covalent cross-linking occurs. So here's a telo domain, and it's binding to the next to form a fibril. Okay, you can buy, when we buy collagen, you know, our little bottles of collagen, most of what we buy is telocollagen. So it will have these domains, and it will be, you know, likely much more complex fibrillar structure. You can buy a telo, and so you're going to have much less of a complex structure.
And that word, can you spell it? Tilo?
T-E-L-O.
Thank you.
Yeah. So I just wanted to give you a sense of the scale of collagen, the different forms of collagen. Again, this is not the greatest slide, but it's sort of hard to find. It's been really hard to find better ones, but you get a sense of going from an amino acid to the tropocollagen, that's the triple helical monomer, to the fibrils, to the fibers. And this just shows you on this side what the length is. The diameter of a fibril is going to be 15 to 500 nanometers, but it's going to vary depending on tissue and context. But that just gives you roughly what scale we're talking about. So collagen cross-linking is very important. As you can imagine, because you don't just have these single chains, you have fibrils that then form fibers. It's very important to understand what these cross-links are, and they determine collagen mechanics in a very big way. So there are three kinds of cross-links. Cross-links from lysyl hydroxylase and lysyl oxidase, cross-links from transglutaminase, and cross-links from advanced glycation end products. The latter, I've always sort of blown off because it wasn't clear what that ever meant. And then I was involved in this paper that showed that really it was hugely relevant. And so I'll tell you about that in just a second. These are all post-translational modifications, okay? So let me tell you, the first one is lysyl hydroxylase and lysyl oxidase. So these crosslinks occur in two stages. The first is lysines are hydroxylated, and there's a family of enzymes that do that. They have different specificities. There's a lot of variability, but they're really important in disease. There's evidence that certain kinds of cancer are more or less facilitated by the presence of different kinds of lysyl hydroxylases in the local matrix. And these lysyl hydroxylases determine the final type of cross-linking. They're all different kinds of this family of cross-links. And then step two is mediated by the lysyl oxidases. There was huge publicity about them a few years ago when the pharmaceutical company Gilead spent like billions of dollars on clinical trials trying to show that lysyl oxidase inhibitors might be, you know, anti-cancer, anti-fibrosis agents. All failed. But so there are 5 family members, LOX and LOX-like 1, 2, 3, and 4. These are copper-dependent enzymes. So people with copper deficiency are going to have abnormalities in collagen cross-linking. And what LOX does, it mediates an oxidative deamination in the telopeptide regions, and that then spontaneously react to form crosslinks. And that's called maturation. I'll show you a picture of that in a minute. But I want to emphasize that this crosslinking causes the matrix, the collagen, to get stiffer. So, and this is going to be very important. So in disease, in general, stiffness is a bad thing for the progression of fibrosis or cancer, for example. And it's often related to increased stiffness because of crosslinking. And so this is a way complicated slide, but I just wanted to, I just want to make a couple points about it. The details don't matter. So this is what lysyl hydroxylase and LOX crosslinks look like. Again, we talked about the telepeptides and how one of the ends of that bond is going to be in a telepeptide and the other will be internal to the collagen. So you get this staggered stance. But so there are a whole bunch of different kinds of crosslinks mediated by lysyl hydroxylase and LOX. They're going to be determined by the specific enzyme, specifically the lysyl hydroxylase. And you can get immature and then mature crosslinks. The details don't matter, just that it happens. And I just want to note that these enzymes act on both collagen and elastin. We're just going to talk about collagen. Okay, so details don't matter, but I just want you to have some sense of the basic collagen cross-linking, and this is a combination of two enzymes. Tissue transglutaminase is different chemistry. Again, there's a family of enzymes. These cross-link many ECM molecules, including collagen, including fibronectin. And like the lysyl hydroxylase, lysyl oxidase crosslinks, it increases ECM stiffness. Advanced glycation end products, these are non-enzymatic crosslinks, and they are associated with high glucose. So these are very common in people with diabetes. They're also, and they can occur inside the body in patients with diabetes, but you also find them in food products. So one of the ways you make these in the lab, and this should give you pause during barbecue season, is you take food and you burn it. That's how you do it in the lab. And the fact is that food that is charred will have more of these, and they're bad. So you do see the increase in type 2 diabetes and also what used to be called non-alcoholic steatohepatitis, but steatotic or fatty liver disease. So the reason these got interesting is that there was recently a paper from Natalie Torek at Stanford, and I'll just say that I was involved in this paper, but what she was able to show was that patients with diabetes or fatty liver disease, as well as experimental animals, when they had high level of ages, they got this very weird cross-linked and bundled collagen. So Natalie had first shown me these pictures about, 5 years ago. I had never seen anything like it. This is a normal, what a normal collagen network would look like. This is what collagen bundles look like when there's high ages. So it completely screws up collagen structure. There is no coherent network. And the interesting thing is that the mechanics of the collagen network completely change. You have much more stress relaxation, faster stress relaxation, and increased viscoelasticity. And in this paper, she was able to show that it led to increased cancer in the tissue she was looking at. So I think this is a pretty cool paper, and I think it's worth looking at if you're interested in collagen structure and how collagen structure can have really profound biological impacts. This paper just came out. And I just want to make a side note that I think we've all been to talks where people put up, a slide that has the headings of all like 8 or 7 of their science and nature papers, with the titles and all the authors. And it's pretty useless for you, right? It impresses you that, this person does a lot of science and nature papers, but it doesn't really help you. And so the Howard Hughes Medical Institute has recently been encouraging people to not do that and to not list journal names and references in the slides, but to give you something useful like the PubMed ID number. And so I haven't managed to convert them all over, but I've tried to do that here. So if you're interested in a reference, there's the PubMed ID number. And anyone who's ever tried to find, a common last name, in China, it might be Wang. In the US, it might be Wells or Smith. And, just based on a date in the last name and maybe the journal name, it can be absolutely impossible. So I think this is very useful. I encourage you guys all to think about that, but that's a diversion. Okay, so collagen is made It's cross-linked extracellularly, and that cross-linking has really profound mechanical impacts, both on stiffness, elasticity, and on viscosity. Okay, yeah.
I think I might have missed it. So the H's interferes with the collagen peptides. But like, does it block them from forming, or does it just make them form really weird?
It makes them form really weird structures. Yeah, it's amazing that it happens, but they actually in their paper they did modeling and showed that this does sort of make sense. What it does is it interferes with those telepeptide mediated cross-linking. So yeah, it's a very nicely done paper. It's pretty complicated, but I had to read it a few times myself, but it's worth reading it because it tells you a lot about collagen structure. And I think it was quite a surprise to people. So I just wanted to talk a little bit about collagen alignment. And so collagen is generally not, sort of thrown in randomly. It is often aligned. Think about a tendon, for example. You have, very, very nicely and beautifully aligned fibrils. You often have it, in ducts, for example, you'll have very nice collagen alignment. And how does that alignment happen? And there are really two ways. The first is in some tissues, collagen alignment occurs at the time of synthesis. And tendon is one of those tissues. These are some beautiful pictures from Carl Cadler's lab at the University of Manchester. And what they were able to show is that collagen secreting cells have these things called fibropositors. And this is a fibropositor, for example, that guide collagen as it's being synthesized and secreted from the cell. And they guide it into an aligned, fibrils, fibers, and bundles. So that is one way. That tends to be seen in very rich collagen-rich tissues, but again, it's not entirely understood yet. There is also post deposition alignment in response to cellular forces. So this is a paper from our CEMB group showing that when you actually when you pull on a random collagen network, you can actually get it to align. And I will show you the picture. This is from Vivek Shenoy's lab. So this is a computational modeling. You can see an aligned collagen crosslink. Now, the key thing is a collagen network. The key thing is that this network must be cross-linked. Okay, and what happens is if you pull on the edges of it, on the ends of it, you can see that collagen will align. A number of these fibers are going to align. And as an example, think about if you took chicken wire in your garden and you pulled on it, each little piece of the wire in chicken wire is pretty short, but they're all effectively cross-linked. And so when you pull on it, you could get, your chicken wire, you'd have aligned wire all through the middle. And this is what happens. This is what happens with collagen, and we're not going to talk about it today, but this is a really important for tissue mechanics. So, but my point is that collagen can get aligned in a couple different ways. It can get aligned at the time of extrusion from the cell or secretion from the cell, and it can also get aligned post deposition. And I just wanted to, again, highlight this issue of length scale. The details of this diagram don't matter, but I just wanted to show you that going from the assembly of the triple helix to, the formation of a mature collagen network, a mature collagen matrix that might be quite complex. We're talking about really widely varying length scales, and that this also occurs over widely varying time scales. So collagen, in many cases, lasts for the entire life of the organism. So much of your collagen was your collagen when you were born, and it's going to stay that way. There is clearly some repair, but the time scales for collagen synthesis and organization are really astoundingly varied, as are the length scales. And just to remind you that the way collagen is organized, we talked about, you have the triple helix, the monomers are synthesized, they form a triple helix, they form fibrils and fibers, and then they organize more into networks and bundles. And that can be normal, or you can have disorganization, as I showed you for that age paper from Stanford. They can be very, very disorganized, and that can be important in fibrosis and cancer and aneurysms and so on. And then the one other thing comment that I wanted to make about collagen, these collagen networks, is that when they're reorganized, it's often plastic. And what plastic means in this context is that the changes are irreversible. So that when you pull on the network, you may have stretch dependent breaking and reformation of weak crosslinks. And so the figures don't really matter, just, you don't want to have text on every single slide, right? But the point is that when you reorganize this collagen, it can be effectively irreversible, or much of it can be effectively irreversible. And the interesting thing is the same thing happens in the plant cell wall. That's work that's recently been published by one of our CEMB members. So just a couple quick things about collagen mechanics. They have really high tensile strength, more than steel. But for family members with non-collagenous domains, some of them might be relatively flexible. So you have a very flexible domain in between a very rigid domain. And so you can get some interesting mechanics that way. Collagen is also piezoelectric. And what that means is that if you apply a stress, you apply force to the collagen, you get a small electrical charge, and the reverse, if you apply an electrical charge to collagen, you get a mechanical response. This is potential as a bioforce sensor, but I just want to caution you that the time and length scales are not very well understood. And I hear people talking all the time about how this is going to explain acupuncture, and I don't think that the science is there yet. Collagen mechanics are also non-linear. When you have linear mechanics, it means if you apply 2x force, you get 2x deformation. If you have non-linear, it means, you have deformation 1 with force 1. If you 2x the force, you might have 7 times the deformation, or 1.1 times the deformation. So the collagen is quite non-linear. And the other thing is that stressed collagen fibrils that under force are often protected against degradation. The final thing I want to talk about with collagen, I think, is the question about collagen and stiffness and tissue stiffness, and what is the relationship? So I think there are two sides to this coin, but the two sides are part of the same coin. The first is Dennis Disher's group has done some really nice work, particularly with the literature, looking at the percentage of collagen in different tissues and their mechanical properties. And for the most part, it falls on a line. So the idea is that the more collagen you have, the stiffer your tissues are going to be. And I think to a first approximation, that's absolutely true. And this is a beautiful illustration of that. However, I told you that collagen cross-linking alters mechanics. And I think that is an added complexity when we think about collagen mechanics and what tissue stiffness is as a function of how much collagen is in it. This is work from Mark Jones's group. And what they did was they had lung tissue. They looked at how much collagen it contained and what the stiffness was. So this is this diagram right here. And you can see there's no obvious relationship between the amount of collagen and the stiffness. And I would just say my group has done the same for the liver. There is just no relationship. But if you look at stiffness as a function of crosslinks, these are in this case lysyl oxidase mediated crosslinks, you can see it's a very nice linear relationship. So it's a combination of the amount of collagen and how that collagen is cross-linked and organized that determines tissue stiffness. That makes sense to everybody? Okay, and I just want to give one shout out to the tissue relevance of collagen deposition. And one of the key areas where you see increased collagen deposition is in wound healing and pathological wound healing, which is fibrosis. And this is, so this would be normal wound healing. You have, this is someone's skin, you have a cut, and you have a scar form, but that's normal wound healing. That's very important. This is some increased collagen, but it keeps your skin together. This is pathological skin healing. This is a keloid scar, and you can see that there is a lot of, I mean, as you might guess from looking at the scar, a lot of excess matrix deposited. This is a liver that is cirrhotic. This liver is diseased, and there's massive amounts of collagen deposited, such that the organ no longer functions. Similarly, this is a lung with fibrosis at the top, a kidney with fibrosis. So these are all diseases where there's excess collagen deposited, and that alters function. This is a picture, I think this is lung. I'm actually not sure. I'm not sure what it is, but it just shows you that this would be the normal organ, and you can see all these nicely pink stained cells, and then you have fibrosis. Not many pink stained cells left, and you can see that architecturally things are going to be quite different. There is one exception to this. Can anyone think what it is? The exception to the idea that increased collagen is associated with wound healing and fibrosis. It turns out that in a fetus, fetal wound healing is dominated not by collagen deposition, but by deposition of hyaluronic acid that we'll talk about in a minute. And fetal wound healing heals without a scar. So it's a very interesting, very interesting altered kind of wound healing. So I'll just throw that out. So, all right, we talked about collagen, talked about structural diversity, the basic collagenous domains, and how that may affect collagen mechanics, and how collagen is cross-linked and deposited and aligned. So there are other fibrous ECM components. I'm just going to give you 1 or 2 slides on each. The first is fibronectin. Fibronectin is what's called a linker protein, and it has multiple domains, like 14 or something like that. Some domains bind cells, some domains bind heparin, which is glycosaminoglycan, some bind collagen, but it's sort of linking everything together in the matrix. Fibronectin is organized by cells after it's deposited. It does not self-assemble. So, so you can end up with some very complex fibronectin assemblies, but that all happens organized by the cell after it's deposited. And just quickly, fibronectin is pretty cool mechanically because you can extend it 8-fold before it breaks. It gets wickedly stiff, but it doesn't break. When fibronectin unfolds, it exposes integrin binding sites and it facilitates cell adhesion. So if you want to think about the lecture you just heard, it's very, when you put tension on fibronectin, you're going to increase cell adhesion. I won't say anything more about that. Elastin is the other big structural protein like collagen, but it provides resilience. And maintains the original shape. You can imagine, people get older, their elastin starts to degrade. And, when you sort of pull on their skin, it doesn't bounce back. So that's an example of elastin not being as functional as it was in someone younger. It's 5 times more extensible than a rubber band, which is a fun fact, I think. And is the other thing is it's a major component of the arterial vasculature. So it's very important in, you don't want your arterial vasculature to expand and then not be able to contract. That would sort of be, that would be bad. Does occur in aging. It also is very important in duct mechanics. So when we think about the ducts of the body, elastin is very important for that. Okay, so glycosaminoglycans and proteoglycans. So glycosaminoglycans are unbranched polysaccharides. Most of them are they're all repeating disaccharide units. With the exception of hyaluronic acid, they're almost all sulfated and they're incredibly negatively charged. So there's going to be an awful lot of water that accumulates around glycosaminoglycans. So proteoglycans are just proteins that have GAG modifications. So there are a number of different families, and let me just, the details don't matter, but I just want to say, so one of the families is aggrecan. You see that in a lot of the big structural components of the body. You can see it has, it's not well, it actually has hundreds of glycosaminoglycan side chains, and that's going to make it effectively a huge molecule. There's going to be a lot of water involved. It's going to take up a lot of space. Some of the glycosaminoglycans are much smaller. They might have one or two proteoglycans are smaller. They might have one or two glycosaminoglycan side chains. There are also signaling receptors that might have GAG side chains, but they're just very important in both structure and the signaling abilities of these proteins. Let's see. Just quickly, they do form a hydrated gel. So we talked at the beginning about, that the ECM is really a hydrated polymer. And so you have fibrous proteins that are embedded within these proteoglycans. It really fills the extracellular space. There's no excess space in the body. And the negative charges attract cations and their osmotic forces. So you actually have swelling. So when you get, when you get bitten by, a bee or something, and you have, your skin swells, that's, water is being brought in, and is causing swelling of the ECM due to, glycosaminoglycans. It's, quite hydrophilic and stiff, and critically, they resist compressive forces. So when you add proteoglycans and GAGs to collagen, as typically happens, it alters the mechanics. And I just want to say, they're very important. Proteoglycans and GAGs are really important to collagen assembly, particularly some of these small proteoglycans. You might have heard people talking about biglycan, fibromodulin, lumican. And they're very important in collagen assembly. This is just an example of cornea and dermis with and without lumican. So these are knockouts with and without. And you can see that the regularity of collagen assembly really depends on these proteoglycans. And you see abnormal collagen organization and assembly with knockout of almost all the small proteoglycans. So lastly, I wanted to talk about hyaluronic acid, or hyaluronin. It is a non-sulfated GAG. It is not protein. It doesn't have a main protein, and it's huge. It drives shape changes. That was the total length can be more than 10 microns. That was, the box that I showed you in my ECM scale picture, very much associated with water molecules. If you look up hyaluronic acid on Google, what you will find are thousands and thousands of ads for cosmetic treatments. Because when people want to plump up anything on their face or wherever, they get injections of hyaluronic acid because it brings in water and plumps things up. It does serve a nidus for the formation of aggregates with proteins and proteoglycans and so on, although it itself is not covalently linked to any kind of protein. Hyaluronic acid comes in many different sizes, widely ranging sizes, and has very different functions. In its large size, it's part of fetal wound healing and leads to scarless healing. In its small size, it's very fibrotic and pro-inflammatory. So, okay. So I just want to give you now two specific examples of ECM assemblies. The first is the basement membrane. I just want to say it's quite variable and also quite anisotropic as far as mechanics. We'll leave it at that. And then I just wanted to, as I said before, tell you a little bit about this glycocalyx. This is this coating on the surface of basically all cells. It's a coating that includes hyaluronic acid, mucins, large proteoglycans, and they're really crowded. And they give you an osmotic pressure gradient as you approach the surface of a cell. None of us ever think about this when we think about receptor biology, right? But it's very important. But this has a lot of implications for how soluble factors bind to their receptors. Very highly charged, and it's been modeled as like a bed of grass, or thought of as like a bed of grass, and modeled with the physics of polymer brushes. So for people who are interested in polymers, and I know there are a few of you here who are, this has been applied to thinking about the glycocalyx. I'll skip this. So just for final notes, I just want to make, I want to make the point that in the lab, we use individual matrix proteins, but this is not even remotely physiological. The forms, the ECM, all parts of it are made of these very large heterogeneous aggregates, and we simply do not study that in the lab. And some of the examples are the small leucine-rich proteoglycans, which I showed you are important for collagen organization. Fibronectin has domains that bind GAGs and bind collagen, and is really important for collagen organization. Collagen and elastin are intertwined, and on and on and on. There are, just aggregates are very important in the body, but not so much in our labs. So finally, I just want to leave you with a couple of things to think about when you think about the matrix. So the water, a third of body water is extracellular, and it's all in the ECM. So water is a really key part of the extracellular matrix. We might not talk about that, but it is. The second is that there are interactions between the cell and the matrix, and they go in both directions. So cells reorganize the matrix, but cells also respond to what the matrix is doing in matrix structure. And it is sort of a dynamic and constantly changing set of interactions. It's important to think about how things move through the matrix, both the interstitial matrix, the basement membrane, the glycocalyx, because it's not just that, your little soluble molecule that I talked about before is zipping along and just happily goes up to a cell receptor. It has to move through a very complex and crowded ECM. It has to move through the glycocalyx. It might have to move through the basement membrane. And understanding how cells and solutes and fluid move through the matrix is something that is very important. There is some work being done on that now, but it is still nowhere near fully understood. The next thing is think about scale. And I've highlighted this many times, but matrix molecules form aggregates, and they may have different behavior if they're individual molecules versus aggregated molecules, particularly if they're heterogeneous aggregates. So this is just something important to keep in mind for you. And similarly, the mechanics of individual molecules compared to the mechanics of tissues are going to be quite different. So collagen, if we compress collagen versus compressed collagen filled tissues, or cell-filled tissues with collagen, there are going to be completely different kinds of mechanics. Tissue mechanics in general is really a function of individual molecules combining to form aggregates, often cross-linked networks with cells present. And you can have behavior that's completely 180-degree different depending on what the organization of cells and molecules is. Finally, I talked before about crowding and how that really affects really everything as far as ECM behavior. And finally, there is an intersection, not necessarily well understood, about the biological and biophysical functions of the ECM and its mechanical functions. We think about how ECM components may be ligands and interact with receptors, but we have to think about the fact that they also have mechanical functions, and they're probably not completely separate. There's probably an intersection. So overall, what I want to leave you with is that the ECM is very complex, much more complex than we often give it credit for when we do our experiments, or particularly when we read other people's papers. So I just want to give you some sense. Obviously, not every detail matters, but I want to give you some sense of the big picture of the ECM. So I will finish up, and if anyone has any questions, please feel free to ask.
I just have a quick question. So when a cell secretes collagen, is all that collagen actually, do they all self-assemble?
I'm sorry.
Or does only, how would you? Yeah.
So it's all gonna, when it's secreted, it's going to assemble,
Oh,
and it will be cross-linked post-translationally.
Okay.
But then it will undergo further organization.
Okay.
And with application of forces, then it's going to reassemble. So it's really dynamic starting at the time of secretion.
Okay.
Yeah. It's one thing that is not very well understood. I mean, we understand collagen structure, collagen organization up to a point, but there is a level of complex organization that really is not understood at all. Yeah.
Usually in a disease setting like fibrosis, when it comes to increasing in elastic modules of the tissue, does the excess deposition of matrix contribute more to that increase, or does the increase of cross-linking?
It's both and it depends on timing. So for example, my lab studies liver fibrosis and stiffness does not increase initially. Stiffness doesn't increase till late, stiffness goes up right away. But collagen doesn't go up till later. And the same thing is true in the lung. That work that I showed you from Mark Jones in lung fibrosis, initially there is no difference in collagen content in those tissues, and yet stiffness goes way, way up. And the thought is that that increase in stiffness is going to be driving further collagen deposition. I think later on, probably the amount of collagen makes more of a difference, but you can't separate the two of them. That's my point. Yes.
So,
Oh, go ahead.
so does the amount of collagen affect the organization of the collagen, especially the orientation? I'm thinking about
Sure,
kilobytes.
yeah, absolutely. You know, if you just have two fibrils and they're cross-linked, you're not going to be able to, you know, say align them very well if you apply force, right? But if you have a large amount of collagen with rich cross-linking, then you're going to be able to get much more complicated structures when you apply force. Yeah. And
Okay, I kind of had a follow-up to his question. So you mentioned that most of the collagen we have, what we're born with. And when you have the stiffening of these tissues, because there's more collagen, is that collagen being taken from somewhere else, or is the
no, it's definitely not. If it stiffens because of more collagen, it's newly secreted. It can get broken down, so fibrosis, particularly in some tissues, can regress, and that collagen will get broken down. But, you know, if you think about your tendon, for example, your tendon, you know, there may have been some more collagen added to it as you know, go through, you know, as you grow up. But the collagen you have as an adult is probably the collagen you've had since, you know, you entered adulthood. There is this beautiful paper, though, from Carl Cadler showing that that particularly in tissues that are subject to very high loads, like tendon, you actually have repair in a circadian fashion. So what he showed is that, you know, we think of tendon, you have these nicely aligned and uniform collagen fibers, but there are also this other population of much tinier collagen fibers that at night, they go in and they repair damaged collagen. You don't see those in every tissue, you know, tissues that aren't subject to high loads don't have them, but I encourage you to read the paper and think about, you know, does it make sense? I think it's actually a pretty cool paper, but I mean, you know, you like, you'd have to imagine that your collagen could get repaired in some way if it was damaged, but yeah. So, but the point is it's very, very long-lived.
For a cross-linked, it seemed like there were like typically 3 points of connections. Can it ever happen that one point of connection breaks and you still have the other two?
That's just the 3 points of connection is just a graphic.
Yeah.
Yeah, there can be multiple points of connection.
And does that drastically affect the stability?
Yeah, it can. Now, remember, for many of these crosslinks, particularly the lysyl oxidase crosslinks, these are covalent bonds, so they're not going to break. Something else will likely happen. There may be sliding but they're probably not going to break. They'd need enzymatic destruction. I don't even know if that can happen, but yeah. But it's going to be variable how many crosslinks you have. Yeah. The illustration was just, you know, for illustrative purposes. Yeah.
A lot of the hydro materials that we used to study, like you know, matrix mechanics and such, so like some of them are nanopores, some of them are like more macropores. So I guess my question is, in native tissues, are there examples of like nanopores versus macropores, ECM?
Pore size in the body is going to be quite variable. I can give you specific examples, but it will be variable. In the lab, we tend to use hydrogels with different porosity depending on whether we want cells to be able to migrate through them or not. So that's sort of the operational decision for what hydrogels you use, but in the body, it's going to be variable.
So when it comes to, I guess a follow-up question is, when it comes to nanoporous gels, do cells just don't migrate in them at all, or does it say?
Yeah, I mean, if there's, cells can squeeze, and I think there's a lecture tomorrow from Min-Pro, so I think you'll hear some of that. Cells can squeeze through very, very small spaces. I mean, the key is getting the nucleus through. But there are limits. And people have done that experimentally, they make, really, really tiny pores or pores of different sizes going down to really tiny and see when a cell can migrate through, and when it just gets stuck, at the entrance. So, and the same is, theoretically, I mean, presumably the same is true in vivo. Does that answer your question? Okay.
Are you targeting collagen deposition in your liver fibrosis model?
So a lot of people are targeting collagen deposition. The reality is that the liver is actually pretty good at targeting collagen itself to a point. Liver fibrosis can regress. And in fact, I don't know if you know, but up until recently, the major cause of end-stage liver disease was hepatitis C infection. But now there are drugs, so we can cure everyone of their hepatitis C. And it turns out if you do that, their incredible matrix deposition in their livers tends to regress. Whether there's a point of no return and what that is questioned. But definitely cells are, the organ is able to regress itself. It's not specifically what I'm working on. It's more in the hands of pharmaceutical companies at this point. But yeah, that is. I think it turns out if you remove the major injury in many tissues, then the organs will heal themselves as far as fibrosis. But figuring out what that injury is and getting rid of it is the big problem. Yeah, I'd say particularly in like lung fibrosis, we have no idea what causes many kinds of lung fibrosis.